Well, good afternoon. My name is Brian Staley. I'm President and CEO of the Environmental Research and Education Foundation, and I'd like to welcome you to our science session today. A couple of housekeeping announcements before we start. Just note that all participants are muted. If you want to submit questions throughout the presentation, feel free to do so using the questions function on your control bar. If you need something, please chat with the organizer. We'll be happy to help you out there. Note that slides are available for download in the handout section of the GoToMeeting app. And then for those of you who are attending this live, you will receive a certificate of completion after answering a brief survey in the follow-up email we'll send from GoToWebinar, and that's going to happen within a number of hours or within 24 hours of the ending of this webinar. So with that, I would like to welcome you to today's topic on black goo. What is black goo? We're going to find that out. I mean, I think there's been a lot of speculation on what black goo is. I'm reminded of some of the old horror movies I used to watch back in the day of the blob coming down from outer space as a sentient entity. Is that what black goo is? We're going to find out today. Probably not quite that provocative, but nonetheless, there are going to be some really interesting results that our team of experts are going to share about black goo. And so I think this is going to be a really fun discussion. So with that, I'd like to introduce our experts and our speakers for today. We have Dr. Craig Benson, who is an international authority on everything waste and landfills. His expertise is rather broad. He has done a lot of work at the University of Wisconsin as well as the University of Virginia, and has a tremendous number of accolades behind that vast experience of his. We're also joined by Brittney Crawford, who has over 8 years of experience in the engineering field. She hails from the region of Waterloo's Waste Management Division and has been focused on black goo for a number of years. And also joined by Dr. Laura Hugg, Associate Professor with the University of Waterloo as well. Has done a fair amount of work on looking at the black goo phenomena from a microbiological aspect as well. And so with that, I'd like to welcome our speakers. I'm glad you're here today. Appreciate you being here to join us. And we'd like to just sort of start off in the sense of black goo. What do we know about it? One of the biggest challenges with black goo, as you can see from the pictures here, is that it creates a huge number of operational challenges. And so we want to get into kind of what some of these operational challenges are. Brittney, I'll go to you. What are your comments about the operational challenges related to black goo?
For sure. Just before we get started, I just want to thank you, Brian, for the kind introduction, and thank you, Environmental Research and Education Fund, for having me as a panelist today. And I'm really happy to be here representing from the region of Waterloo, and I'd like to acknowledge the team there, the folks at Geosyntec Consultants and Serum Labs for all their contributions to this project. So to get back to your question, what are the challenges? Well, you know what? There's a ton, because this is a great it really gives context for the scale of the issues that we're seeing at numerous sites and kind of how ubiquitous this problem has now become. Sorry, could you go back up to the first slide? Yeah, so I just wanted to showcase a lot of these photos that you can see where there are several maintenance issues happening, and then as a result of this black goo formation, operational issues happening. So these photos are taken from various different landfill sites, and unfortunately, there's quite a few in there from the region of Waterloo. So as you can see on your screen, you have on top left dual phase extraction well clogging. So this not only affects the landfill gas, but also the leachate collection system. There's leachate pump screens and intakes that are clogging. There's inaccuracy with transducers. We've had a number of transducers fully wrapped with black goo. Then you have vac hoses clogging. So you can see a mass amount in the top right that's stuck onto the end of a vac hose. And then in the bottom right, you can see a contractor pulling this black goo that's come up from the bottom of one of our wet wells. And it really showcases the cohesive strength and not only the tensile strength of this substance, which is pretty wild to me. Another challenge that we experience is it's really kind of unpredictable. We've tried to map it at our site. We have an event timeline, but it will lay dormant and then it will show up again. So we haven't really figured out the rhyme or reason or really what is going on with this substance. And to bring that all together, all of this leads to challenges with leachate and landfill gas systems not operating as originally intended. So you have to decrease flow rates, you have risks of pumps failing, screens fouling, and pipes clogging that are unfortunately inaccessible, which is a big issue and I think an issue for a lot of landfill operators. And this leads to increased maintenance at prone locations, high operational costs, and honestly just increases the probability of failure of assets. All right, next slide. Thanks, Brian.
Sure.
So I really wanted to there's some of you who might not know what black goo is, and videos tell you everything. And as you can see here, here's a pretty good visual representation of what this mysterious substance really is. We call it black goo because that's just what it looks like. So our pump intake screen, we had some flow irregularities. We pulled the pump and it's no explanation why that screen was completely clogged with this like really membrane material. That was interesting when we first found that. And then the video that's now playing on the right, that was a sample that was collected from one of our bottom of our leachate manholes during a flushing event. And that had enough strength to stay on the end of a back nozzle, which is pretty astonishing. So overall, it's a very big issue for landfill operators, and that's just my take.
Thanks so much for that, Brittney. Craig, what are your thoughts on the operational challenges as well?
Well, it's a mess, that's for sure, when you're running into black goo. I think characterized it well. And in some sites, it's really a significant problem. The one on the right here is a dual phase extraction well pump that had been in service for 6 hours. 6 hours, right? It's completely covered, it's non-functional. And you see some of the other examples here where just the goo is around pumps and clogging pipes, screens. At some sites that I've been dealing with, they're cycling pumps out every day or every other day and rebuilding the pump. They have essentially 2 sets of pumps, one to have running and one to rebuild, cycling them in and out. I think this slide, along with those that Brittney showed, really gives you an example how difficult this stuff is. Black, sticky, rubbery, also smells terrible. And then you see on the right just how sticky it is. And so that's getting stuck in pumps and pores, check valves, you name it, it gets stuck there.
So Craig, when we think about where we're seeing this, and you all gave a great description of kind of where it's showing up within the landfill itself, but maybe give us some clues as to what zooming out of the landfill and thinking of this more from a geographic sense, or maybe even weather regimes, like are we seeing this black goo in specific geographical areas, or is there some sort of pattern that you're kind of noticing that's evolving as we're starting to detect black goo in these different sites?
Well, one of the things that we've observed is it tends to be in wetter areas. And I would say even we're a little more general than that, at wet sites. I actually was on the phone the other day with somebody in California at a site where they had done a lot of recirculation, not a wet location, but a lot of recirculation at that site, and they've got black goo problems. So my first experience was a site that was watered in. We had issues with leachate extraction, with odors, and at that site, all the dual phase pumps were getting clogged up on a regular basis. You can see here in this map, there's 30 different sites around the US, predominantly MSW landfills, but not exclusively, but also as we know in Canada as well, seeing the same types of things. Also in Puerto Rico, we have a sample from Guam. We get a sample from Guam. That was really pretty cool to be able to get that. A couple of weeks ago, I was on the phone with somebody, and then on a video call with somebody from Brazil. They've got the same issues at a site in Brazil. But the common thread is a lot of moisture. And I think the moisture is one of the things that helps the goo move through the system and then collect in places where we don't want it to collect.
Now, so given what you just said, seeing the absence of detections really out to the western part of the United States, is that because that sites have not been sampled and so therefore we don't know if there's black goo, or is it because sites have been sampled and so far very little black goo instances have been observed?
Yeah. I don't know the answer to that for sure, but what I suspect is that they're just not seeing it at those sites because they don't have the problem. They haven't been going in and looking for it, but if they had it, they would know about it, right? And we've been getting samples from all over, and I haven't had anybody calling me from Denver, for example, hey, will you look at this black goo for me? So I suspect in drier areas we have less problem just because we have less leachate moving within the waste mass.
Gotcha. Well, yeah, you made a great point there. I mean, if you have it, you'll know it, because given some of the pictures that you all have shown so far, it seems like it would be a hard thing to miss for sure. Brittney, let's go to you. What are your thoughts on how things have progressed in terms of just sort of identifying black goo and where you're seeing it?
Yeah, I wanted to tackle this based at our landfill site and specifically where we're seeing it in the landfill. Craig did a good job giving the overview because we originally thought, to be honest, we had a bunch of theories going, oh, well, it's because we removed organics, so the waste is breaking down in a different way. Well, no, that was debunked because Craig said there's landfills in the States that do accept organics. So a lot of these theories were debunked. So I'm not sure about the specific types of landfill because those where leachates recirculated is happening. So at our site, we've had 23 events since 2012, and this event timeline has been pretty good to map prone locations. Typically, we do see it on our pumps in our leachate wells and in our leachate collection system. So it first appeared in December when we pulled the pump for annual maintenance, and we had this mass, as you can see. And then I lay dormant or maybe undiscovered. I don't know what was the case until 2017. And this was a major operational event at our site where a condensate check valve was clogged. And this was a big moment where we had to stop because there was surging in our header. It caused major operational issues because we have a gas utilization system. Tormont's downstream, we had to slow them down. So it's just, we want to shy away from that. And yeah, and then we found this goo in our landfill gas system, which was pretty astonishing. And then from there, during annual flushing is where we see it quite a bit. So at the bottom of our collection manholes on our perimeter gravity drain system. And then this finally, this is a new pump station that just went in. So it was commissioned early 2021. And this was the first cleaning at this location, and there was 2 to 4 inches that had already grown on the bottom of the inlet chamber. And this video really is to showcase that high pressure jet nozzle was not enough to break up this material. It was just caked on the bottom. And in order to break that up to clean out that chamber, we had to go through a whole confined space entry protocol, which we don't want to do. But we've had to, and we've had to up cleaning at that certain location.
Okay.
It likes the bottom of pump stations. We don't have any dual-faced wells at our site, but adhering to transducers, pumps, and pump chains. That's where we found it.
What strikes me is just that, like you said, a high pressure hose isn't really touching it. That's just amazing. It's not really pulling it off of the floor of that pump station. That's really phenomenal. So that kind of begs the question, what in the world is this stuff? So Laura Hug, let's go to you, and maybe you can shed some light on what this stuff is.
Sure. So I am a microbiologist, and when I see strange amorphous globby structures, I just instinctively and immediately think, this is microbial. This is something that's growing in the system. And I think that's a theory that's been debunked. But when Brittney first called me and said, look, we have this weird goo, can I drop you off a sample? Of course I said yes. Who wouldn't want to work with something like this swamp thing? And so we have been looking at it from the perspective of, we know there are microbes in and throughout a landfill, that they interact with all of the waste in different ways. And we've been working with this goo from the perspective of, who is in there? What are they doing in that system? What are the activities? Are they contributing to growth? Can we identify who would be creating this sort of viscous substratum and shape of this biofilm? So if you go to the next slide. And one of the theories was that if this was microbially generated, then there should be, oh, and I think that these are Craig's actually. So I think it makes sense if Craig tackles this first, because I'll tell you, my initial goal was that this was probably microbially generated, that this was something that you could inhibit or prevent from growing by addressing the organisms present. And then we started partnering with Craig, and I think that very quickly became quite a different lens on what the role of the microbes is. So I think I'm going to let Craig talk about what it is, and then we'll talk maybe again about what the microbes are doing.
Okay.
Yeah, glad to do so. Brian, maybe just bump back one slide. Oh, there seemed to be one in between there. Maybe not. Okay, we'll start. This is good. We started looking at what these were from a number of different perspectives, but one of the tools that we used for similar types of materials that we had been investigating previously are tools that look at molecular structure, like FTIR, or Fourier transform infrared spectroscopy. That's a big word. Things, nuclear magnetic resonance, and then would use thermal gravimetric analysis as well. But all these different tools point to different molecular structures, and we can look then for similarities in the molecular structure of the different goos from around sites that we've collected samples, and for other materials like them that we run into. And one of the things that we found is there's a remarkable consistency in nearly all the goos that we had in our laboratory. Remarkable consistency. They all look from a FTIR and an NMR and a TGA perspective to be essentially the same thing, which to me, we're collecting them all around North America, Guam, Puerto Rico. How can that be, right? All these different locations, and they're all about the same. And then we start, what's neat about some of these devices we have, is you can look in a library. They have a library of spectra, and you could ask the device, please go match this with something that you know about. And so we went and began to look at libraries, and what we were found is that consistently we would find polyacrylates and polyacrylamides, typical hydrophilic polymers that are in our everyday life. All of the goos too were tended to be very wet, which is consistent with a hydrophilic polymer. It likes to bond water. It likes to hang out with water. One of the things that we found consistently too is that things that were goos always had an organic content they had high water content. We'll see that in a bit, but an organic content as well of at least 50%. So they're binding organic material, but the goo itself, if it's a polymeric material, is an organic material. So at least 50% of it was organic.
Gotcha. So then I think this next slide here talks about the water content, right?
Yeah. So this slide, and you can just click through the animation a little bit here, Brian, but consistently, goos, because they bind water, they have high water contents, at least 200% on a gravimetric basis, right? So it's got a lot of water tied in with the other solid structure. So that's another tool that we can look at. It's very consistent with what we see with hydrophilic polymers. They bind a lot of water because that's what they're supposed to do.
Gotcha. Okay. So narrowing it down a little bit more, and I think Brittney, you did a little bit of work on this too in evaluating the black goo problem. Do you want to share some of your experiences on that?
Yeah, this is definitely from the lens of a landfill operator and an issue at our landfill, and taking that operational perspective on it. So we realized really in 2017 when our gas system was affected that, hey, we have a major issue. And at the time, we really thought it was isolated to our site. I hadn't heard from others when I reached out, but I think everybody was like, whoa, what is this? We're really trying to figure it out. And then now we've all come together, which has been pretty nice. But this really triggered the need for a long-term treatment strategy. And at our site, we do not have on-site treatment of leachate. It's sent to a wastewater treatment plant downstream. And we knew long-term, that something treating this probably would look like a chemical treatment. We also didn't know what we were treating, right? So we knew that we had to identify this and characterize it. And so in order for us to, one, have confidence in what we were deploying into our system, and two, know that we weren't going to alternatively affect the downstream receiver, we knew that this would require bench scale and pilot studies. And then we knew we needed a solution immediately, right? Because we needed everything to flow through the system and not clog and cause any further issues. And just from practices, our existing treatments did work. So we were just going to leverage those, increase flushing at prone locations, just really monitoring. We trialed a hot water flush, which I'll get to a little bit later on, but that's what we did in the interim. And then next slide. All right, so for tackling this long term, we knew that there would probably be a phased approach to this. And that's when we brought on consultants to assist with this, Geosyntic and Serum. And at the time, and Laura's probably going to get to it, we really thought this was microbial based. So when we were looking at tackling it, it looked something like a biocide or a biodispersant. And so we ran some bench tests, which were pretty favorable, but there was no really smoking gun and clear definitive that this would work. So from there, we said, well, why don't we, the best way to do this, and because once you put something in the leachate or into a landfill, you really are unsure of what's going to happen. I mean, you just know operationally that things are working, but you don't 100% know what's going on. So we wanted to trial a pilot test system where we trialed amendments. And with that, figuring out, is there a treatment solution that works for our site? And ultimately, then we would get a recommendation and deploy it. So along the way, I do want to mention, over the years, we've collected samples and we've sent them to Craig's lab and Laura's lab and Serum's lab for analysis. And that's really helped informed us as we've tried to figure out what this black goo is.
Okay, and so we've heard terms used so far like this is growing here. Craig used the term polyacrylamide, which does not sound biological, but yet that kind of begs the question. I mean, is this biological or is it abiotic or non-biological? And maybe there's not quite a clear-cut answer in some respects, but maybe a clear-cut answer in other ways. And let's maybe get into that. Craig, I think you have a few things that you all have observed in the research that you've done, as well as Laura's group. So why don't you kick this off here? Yeah, Craig, if you want to jump in on this.
Craig, we've lost your audio. I can see you talking, but we've lost your audio.
Okay.
I'm back.
You're back.
The guy with the lawnmower was outside my office window. So I had to go on mute for a little bit. What's the likelihood of that happening? So thank you. But I'll talk to this because this was a question that had been kind of nagging at us. Is this abiotic? Is it biotic? And it's probably somewhere in between. If we look, and so one of the things we did is examine the characteristics of biomass that Laura had made in her lab and compared that to what we saw with the black goos and with some of the similar tools that we have been examining. They're both dark in color, but they're very different materials when we look at their structure. They tend to be dark in color, they have odor, the goos tend to be more sticky and stretchy than the biofilm, but they're not necessarily mutually exclusive. And maybe we go to the next slide, Brian. So one of the things we do is use these tools like FTIR and NMR, and we see that the biomass is very different from the spectra for a goo. The biomass here is shown in purple, and then the other spectra for goos are shown in the other colors. They're distinctly different, suggesting that the two are very dissimilar in their molecular structure. This in itself doesn't necessarily mean that there isn't a biotic process coupled in with an abiotic material at the same time, and I'll touch on that in a minute. But you can see they're distinctly different here. And if we go, I think, to the next slide, Brian, if we do thermogravimetric analysis, which is a fancy way of saying we heat it up and we break it down in an oven. But by doing that, you can look at how the weight of something decreases over time as it decomposes under energy imposed by heat. And for synthetic polymers, we know that they will tend to break down or decompose very rapidly around 500 degrees centigrade. And we see with the biomass, it degrades at a much lower temperature, which is much more consistent for biological polymers. So very distinctly different in TGA. They're two different materials, which suggests that the bulk material for the black goo is an abiotic polymeric material, synthetic polymer like polyacrylate and polyacrylamide that I talked about before. But one of the things that we've been talking about as well is that when you look at pure polyacrylates or pure polyacrylamides, and we have them in our lab that we bought commercially, they don't have that same sticky kind of rubbery appearance. And one of the ideas that Laura has proffered is that maybe that microbial community is interacting with the surface of that goo, creating that real interesting and difficult, challenging rheologic structure on the surface. And that's one of the things that we want to be able to explore.
Well, that's great. And that's a great segue over to Laura, some of the work that you've done. You want to talk to us about whether we're seeing a biological component come into play with the black goo?
Yeah, so one of my initial assumptions was that we would see the same organisms across multiple different goo samples if what was going on was that specific organisms were forming this matrix, were creating the goo, and then other organisms, most microbes like to live on surfaces, were like, hey, here's a new surface, I'm gonna come in and colonize it and go about my daily life, as though I was attached to a refrigerator, attached to something else, right? But those organisms that create biofilms, we know what that entails, we know what that looks like. And so my team went looking for who was shared across these different samples. And so what you're looking at here are some Waterloo goo samples that we had the chance to really do a deep dive into. And so we didn't look at just who was there, but we also looked at, what are they genetically capable of doing? What's their blueprint say that they could be doing at the site? And then also from that, what are they actually doing at that time and space? And then more holistically, what were the total metabolites that were present? So what are the major cycles of functions that are happening within this system? And part of the idea, and certainly my assumption, was that there would be pretty strong similarities across the board. And that wasn't true at all. So, so what you're looking at, the bar charts are just to give you a sense if they're sitting next to each other, they're from the same sample. We just looked at them two different ways. And what you can see is that this is the same site. This is samples taken within the same sort of a 2.5-year span. And there are pretty dramatic differences. This is looking at it from a phylum level, which is the biggest branch on the tree of life. So your main big lineages of organisms. And we see very big differences and shifts within the communities there. And then when we look at that at a more granular view, which is the bubble plot on the other side, you can see that each column is a single sample, and there's very little overlap. These are our most abundant organisms. These are the ones that are there that are dominant in the sample. You don't see the same organism all the way across. You really see kind of patchworky distributions. And again, that was surprising, but sometimes microbes are very happy to swap out who is doing something as long as there's like a shared function. But then when we started collaborating with Craig, we had the opportunity to look at this on a larger sort of geographic scale. So this is the variation that we were seeing in one site. And now we're looking at the variation across geographically distinct samples. And so we have samples, these from Wisconsin, Milwaukee, Florida, Guam, et cetera, are here. Anything that is starting with an ROW or a GS are all the Waterloo. So those first 10 kind of over on the left are all from Waterloo, and you can see the variation there. And again, these color blocks are those phyla, so those really big branches on the tree of life. This is not a detailed look at a single microorganism. This is a big, big group of organisms that have shared characteristics. And you do see pretty strong changes both between sites and geographic changes, and then also within sites. We have multiple samples from a couple different landfills, and they show a huge amount of heterogeneity. And so that's tough. We have 2 organisms that we've identified that are present across all samples, but they range in abundance from 30% of the sample to like less than 0.1% of the sample. And so it's hard to look at those and say, look, that's our smoking gun. These are the important ones. They're consistent though, and that can be meaningful. So the next question was just not who's there, but what are they doing? And when we look at the proteins that these communities are expressing, what we find is that they're very active in methanogenesis. That was very interesting from our perspective because they're making methane while embedded in goo in a methane capture system where the atmospheric methane content is 70%. Like, this is not a thermodynamically reasonable way to be making a living. The methanogens already kind of buck that trend. So what you're seeing here are some different pathways. So methanogenesis is this MCRA gene, and if something is a dotted line, it means the community as a whole has the capacity to do that. We can find it in their genomes. If it's a solid line, it means we actually detected the protein. They're really actually doing that thing. And so we can, and then the weight of the arrow is the amount of that activity we identified. So there's a lot of methanogenesis. There's a little bit of methane oxidation or methanotropy, so eating that methane. And then when we look at nitrogen cycling, this community could do anything with nitrogen, but they're not doing it. They have the ability, but they don't have the activity. And then we see a really strong dissimilar trace sulfate reduction response, which is probably contributing to that black color, getting some iron sulfides, and also to the smell that we get out of the goo. So we can see that like a polyacrylate is a pretty neutral, inert system. A lot of the characteristics of the color and the smell and things like that are certainly coming from the microbes. And I think it's quite an open question right now as to whether or not the microbes are interacting with that polymer, whatever it is, to change those tensile strength, like to get that stretch, that stick, that really goo aspect of it, as opposed to being more of a gel or a flocculate sample. So that's where we're headed next, is to try and figure that out. The trick is that there aren't that many organisms that interact with polyacrylamides or polyacrylates. We don't know who to look for. We don't know what the gene is, and we're gonna work on that in the lab. And then the last piece we did was we looked at that total metabolite data. And we can, like, if there were no microbes in this system, you wouldn't really see metabolites that look like this. You'd really just see the byproducts of whatever that compound was. And we have a pretty rich number. These are the ones we could identify, which is about a quarter of the total metabolites. So this technique is just catching up to the complexity of a system like this. And I just want to draw your attention over to the bar plot on the other side, where I've broken these metabolites up into classes. So benzenoids, organic acids, etc. And you can see that really the dominant metabolites in this system are bisphenol A in blue, the carbonate ion in purple, and palmitic acid. Bisphenol A is a common component of plastics. Carbonate ions can be used as a currency within a microbial community, and palmitic acid is sometimes a byproduct of polymer degradation. And so there's hints here as to what's going on, but I will say that like the 17 most abundant metabolites, we don't know what they were. They didn't match anything in our libraries of metabolites. So we know there's a lot else going on that we can't quite see yet. So there's still some big open questions. And I think, is this goo formed by microbes? No. Is it modified by microbes? Almost certainly. And to what extent and what meaning that provides is what we're asking now.
Wow, it almost seems almost obvious the way you explain it, that the black color that is kind of characteristic of goo seems to be derived more from the microbial activity. Otherwise, you might notice that the goo is more clear, gel-like substance that would be common of like a polyacrylamide or something like that. So very interesting. And that, in that sense, that kind of leads us into what waste streams might be contributing to the black goo phenomenon. And so, Craig, I know you've looked into some of that, perhaps at a backdoor barbecue, even about where some of these kinds of materials may be coming from that end up in a landfill and contributing to the problem. You want to talk to us about that?
Sure. These hydrophilic polymers are ubiquitous in our modern society, polyacrylates in particular. The one in the lower right-hand corner, that's from the steaks that I was cooking on our Memorial Day weekend barbecue. And I took a look, I said the pad on it, and I looked at it carefully. I looked up the brand, I did a little Google search, and it's a polyacrylate because it binds lots of water that would be emanating from the moisture in the meat that I was cooking. But they're ubiquitous in our waste stream. If you've got young children, you're using these regularly for wastewater control. The diapers, meat pads, hygienic pads, all these use these hydrophilic polymers. Another common one is dewatering agents, and something in landfills that we use frequently. We want to get something to pass a paint filter test. We can add something to it that will bind the water and change it from behaving like a liquid and not passing the paint filter test to behaving like a solid and passing the paint filter test. These are some of the materials that we use to blend with wet waste to get them to pass the paint filter test. Not surprising that we would then see them in our landfills and our leachate collection systems. But just a plethora of different applications. One that I've run into that really surprised me is that gas well drilling contractors, when they run into difficulty drilling through wetter waste, will often use some of these polymers to firm up the borehole. Well, we're putting these polymers right where we want to pump out the liquid and the gas, right? Maybe not such a good idea, but they're using them for stabilization. Nobody did that on purpose. It's just, this is something we normally do in drilling. It works in earthen materials. That's where we use it in waste too, but potential problems in those cases. But there's just a lot of different sources of these materials. And over the last decade, and just a comment back from Brittney's slide earlier, she talked about first seeing it in 2012, which is about when I saw it too, 10, 15 years ago. And the volume of these materials, or the total mass used in our industrialized society today, has increased dramatically in the last 20 years, really has. So not surprising in our waste stream. One of the things that we did, if this was truly a polyacrylate or something like it, you should be able to dry it out and then rehydrate it so it would become a solid, and then it would rehydrate and form a gel again. And we did some of that in our lab, and that's what this slide shows. The dehydrated samples on the left, and then we rehydrated it just with deionized water. And what was interesting, the black material was like a skin peeled off, and on the inside was clear jelly-like material, a lot like polyacrylate, right? It was pretty remarkable with this skin, which is probably just reduced it's sulfides, it's iron sulfide or something like that on the outside from the reducing environment, with the inside predominantly just the hydrated goo, the hydrated polymers.
It makes me wonder how many people paused eating their lunch as we go through these slides.
It's good, right? This is just a couple more examples of that same material that we just rehydrated, and we were able to separate that skin from it. Look at the jelly on the left, and then the hand on the right. Make sure you're wearing a glove when you do these things. It looks like a hydrophilic polymer, right? Very similar. And these types of observations that kind of drove us down this path of looking at polymers in the waste stream.
Right. Wow, that's amazing. And Brittney, from kind of the landfill perspective, what's your take on this?
I mean, Craig did a great job. I'll just, I guess, summarize quickly. I mean, if you're an MSW site or you high urban and residential waste streams, you're going to see this. We haven't done any work to really define that in our waste stream. It is quite difficult to do and be open to figure out how to do it. I don't know how much value add there is for that, but yeah, we're seeing every landfill I know kind of in Southern Ontario, unfortunately, is seeing it. Unless you're in urban, actually, there are some rural centers that aren't seeing it so much. So that really goes to show you maybe it definitely is in the MSW.
Okay. Well, now that we kind of described, kind of gotten to the root of what this black goo is, and all the work that you all put into sort of figuring out some of the characteristics of this stuff, it then kind of boils down to, well, how do we remedy this? What kind of methods are being used to remedy sites that are fouled with black goo? Any thoughts on that? Craig, let's start with you.
Well, one of the things that we know when we deal with polymers like these is and you can see they're sticky and viscous, they're not going to move anywhere very easily. So we have to change that, right? And we know with other types of hydrophilic polymers like this, we can change the chemistry, and that will change their rheology and their morphology and create something that's much more mobile. And so, one of the things you can do is add acids. For example, this is one here that was shared to me by a practitioner at Waste Management that they've used successfully as a way to essentially flush out their sumps. This is an organic acid. We've been exploring just using hot washes of concentrated salt solutions, because we know if we bring up the ionic strength, that will tend to cause the polymer to coil and make it more mobile. And as we bring up the temperature, the viscosity of the polymer tends to decrease. It just becomes a lot more fluid-like. So things that we can do to get it out of that viscous, sticky state and into something that's a lot more mobile.
Interesting. And so, Brittney, I know you all have done a lot of different things on interim treatment strategies. Do you want to describe some of your experiences in this realm?
Yeah, for sure. For all the landfill operators out there, we had to look at what we could do reactively to it, and that started with increased flushing at prone locations. And we also retrofitted existing infrastructure just to enable ease for that maintenance activity. So whether it's bringing back hoses all the way down to the bottom, clips at the top, so it's very quick and time efficient. Like Craig mentioned, we did do a hot water flush, and one interesting thing that came from that, it was on one of our sensitive pipe runs where if it did clog, knock on wood, it would be bad, but it picked up the slimy, that slimy kind of material in the black goo, where we had not had that before just with normal flushing. So that's quite interesting. And then again, just monitor flow and pump cycles and just for any irregularities. Okay, so I want to touch on how we have evaluated a long-term treatment plan and what that looks like. And you can see on your screen, this is a CCAN container that has been retrofitted, fully engineered to be safe on site and safe for use. This is what Geosyntec had designed and is currently operating it, and Serum's running the lab analysis. So the intent was to mimic our landfill conditions, our leachate collection system. So it's actually a closed loop system. Leachate is pumped from an adjacent wet well into influent totes where it's sent to 6 reactors that have the same retention time and slope as our leachate collection pipes. And then it is sent back to an effluent tote and then back into our wet well. The whole intention here was to trial amendments. So in theory, we wanted to grow this black goo and then treat it and see if we could grow it. And then next slide. I know this is very dense with information, but if you want to refer back to this, I think it's really valuable. So I'll just briefly go through it, but we were able to grow the black goo. We seeded it. We seeded each of those reactors, and it grew at a rate that we were kind of surprised by. So then we wanted to figure out what conditions were favorable. We then determined anaerobic, which is in the surprise. And since at that time in the project, we really did think that it was rooted in a microbial community. So that's where we looked at a biocide and a biodispersant. And then we upped the concentrations, mixed them together and did a batch and trickle dose. And this is where we started to have funky things happen, actually. There was a solid orange matrix that formed, and I'm happy that it happened in a controlled environment. And that was the whole intention with this pilot program. It completely clogged one of the reactors and we had a spill. So our whole theory is make mistakes in a controlled environment, because if that were to be down to one of our pipes, that would not be great. So then from there, and this is really where we got on board with Craig, where the polyacrylate theory came into play. And our previous treatment phase looked at an industrial cleaner, and then as well as sodium hydroxide and a pH adjustment. And from there, there's been not really a smoking gun per se, and that's really what set us into this last treatment phase. And Craig has mentioned that safe acid, so we are trialing, and it just started in August, the AC 6400. We're also trialing a product called goo-renew. Found out about this at a conference. And then this last one is actually very interesting. So Turgazyme, and we happened upon it in the lab. So when Serum was cleaning in their instruments, the goo was just coming off. So we thought, why not try this in this last phase? And I just really want to mention the importance of lessons learned that we've learned along the way. and how it's really informed the next phase and the collaborative efforts that we've had. And just next slide. And this has been a big thing. There aren't many projects where you have private sector partnering with multiple academic institutions, other public sector entities, and it's really helped, I think, expedite the process of finding a solution, but also, yeah, we can find a treatment, but we want to know what we're treating, and that's really the long-term goal, and to have confidence in that. So it's been great working with Laura and Craig and the folks at Geosyntec and Serum.
Well, and Brittney, let's continue with that kind of thought process, because the whole collaborative nature of this to try to solve the problem, I think is fantastic, and it just shows how when great minds get together, you can really tackle the problem well. And that kind of brings the question into what are the design considerations that allow for a more efficient management of black goo from an operational standpoint? Is there anything that an operator can do that can help at least minimize the formation of black goo in your mind?
To be honest, I mean, if you're dealing with an issue where it is at your site or you're building out the rest of your site, but if it is at your site, ease of access to locations for flushing and cleaning. Unfortunately, I've spoken with other landfills, and they've kind of filled themselves into a situation where they don't have access to certain areas that they might need to clean. So I think primarily ease of access and retrofitting existing infrastructure so you don't have to have confined space entry. And just as Craig was talking, it made me think, well, maybe it's not design, but it's how it's constructed. Do we want to use these slurries? And another thing to mention is some considerations operationally. What are you using for your daily cover? Well, if you're using an alternative cover source, you might be spraying some polyacrylate. So you think you might be helping, but it also might be a hindrance as well.
And Craig, what are your thoughts?
Well, one of the things that I'm just thinking about as we're talking about goo here and looking at some of the photographs is how we keep things moving. You know, one is we want to get access, but we also want to keep the leachate moving. I think about that sump earlier where the leachate is coming in and it's sitting in a sump and the goo is forming along the bottom. We see this in leachate collection systems, in sumps in cells, quiescent locations where these polymers tend to be dissolved in the liquid or flowing with the liquid, and then they get to a point where they're in a quiescent environment and they settle out and they essentially congeal together and form a thick, sticky material. So if it's quiescent, it's going to build up. If we keep it moving, our chance of keeping it out of the pores and in our pipes and flowing out of the system is more likely.
Gotcha. And you briefly touched on this a little bit, Craig. I'm curious to know like why, and Brittney, you as well, but you all said you started seeing this in 2012, kind of like as the first observation. Why are we seeing this now from 2012 till today? Why didn't we see this prior to this time frame? You want to take that one, Craig, first?
I wish we all knew the answer to that, but I think it's one of the things is we, at least in the US, and I'm not sure about Canadian practice, but at the end of the 90s, we started adding a lot more water to landfills. And we were less focused on dry tomb and more about getting moisture in to stimulate biodegradation. And we ended up with sites, a lot of wet sites actually, unintentionally. And I do think the prevalence of a lot of water, wet sites, and these materials in the waste stream is a causative component to this. And in the 90s, sites tended to be drier, and we just use a lot more of this stuff. I mean, you look in just in the industrial literature, the mass produced has gone up a lot, and that mass is going into our landfills as well.
Right. To your point, I mean, I think this material, polyacrylamides and related compounds, seem to be more ubiquitous in society than before. That seems to all make sense. So what happens next? I mean, what does the future hold? Brittney, curious to get your thoughts on that.
Yeah, so our next steps are finishing up the treatment study and then having a recommendation for what we're going to do and deploy it on our site. Serum is doing some sole source carbon plating, which their great minds could speak to if anybody has any questions on that, and as well as doing some mass spectrometry with the University of Toronto. And this kind of just, as that polyacrylate theory came up, we've really kind of pivoted our project scope to then kind of find a solution based on that theory. And it is proving well for us. And then honestly, just continuing our collaborations with industry and academic partners. Again, the collaboration has been huge, and I think that's what landfills will be landfill operators will be happy when we find a solution for this.
Yeah, that's for sure.
Yeah.
And Craig, I'm curious, I think you've done a bit of work on this. Is it possible, you think, to sequester black goo within the waste mass? Is there a way to keep it out of the sumps and the gas wells and everything that's causing problems?
I do think so. You know, if we strategically think about how we fill and how we build cells and fill cells, we can create ways to essentially sequester it in the waste to keep it immobilized. To me, that's the ultimate solution, is to keep it out of the leachate collection system rather than having to clean it. Now, that's easier said than done. We've been building columns in our lab. We've been spiking them with polyacrylate, they're waste columns. We've generated black goo in our leachate collection systems in our columns. So we've shown that we can mobilize it, we can create goo. Now we're exploring how can we build in different essentially filling strategies to isolate the goo within the waste. That's information to come.
Gotcha. Okay. Well, this has been great. I do want to acknowledge you made the point, Brittney, about just how much teamwork is needed to pull all this together, and these are just some of the folks on, I believe, your team that's worked on this and have really provided a lot of insight, as well as a lot of the work that's been happening. I think is this Laura, your group, right? And if you all want to touch base on any of this, feel free to do so. I know with the UW Madison's team as well, it's more than one person. There's a lot of different people who are contributing to this specific effort. Did I miss anyone on the acknowledgement side? I don't want to overlook that for sure. I mean, I know there's a lot of important work going on.
Only one I would add, Brian, is just all the industry collaborators. I mean,
Yeah.
Getting these samples from all over North America and having the opportunity to analyze them. Of course, everybody wants to know the answer, and they've been patient with us as well as we try to figure that out. But this wouldn't have been possible without everybody jumping in and contributing.
Gotcha. Okay, that sounds great. So with that, I think we are at the point where we can get some questions from the audience. And if you have some questions, please let us know, because we'd love to have our group of experts go ahead and address some of that. I'm going to stop sharing the screen here with the slides so that we can keep that focused. We did have a question come in that says, what strategies have you put in place to ensure that non-governmental organizations in developing nations are part of the understanding the issue? Is there anything that you all are doing kind of internationally, whether that, I know we talked about Guam and Puerto Rico, kind of U.S. territories, but I'm just curious to know, have you all done anything internationally beyond North America, or have you even seen this challenge outside of the North American realm?
One example I can share, Brian, is this in Brazil, and it's not something I solicited. They just sent me an email today, and then we had a Zoom call. But yeah, we're seeing it, but we haven't collaborated with them per se. But I do think this issue is in any industrialized society that's got MSWs dealing with this, particularly in wetter parts of the world. So lots of opportunity.
Okay. Brittney, Laura, anything from you all?
I work with a number of different regions in Ontario and look at landfills, but I'm mostly looking at cover soils and leachate. And I just know that in Ontario specifically, a lot of our older MSWs, they don't have really any infrastructure. So it's entirely possible that, you know, some unexpectedly large proportion of the matter below that mound is goo now, but it's not moving, it's not clogging, it's not coming up as a problem. We haven't done a global survey of where we see this. It's more been that, you know, people see that one newspaper article or they talk to us at a conference or something like that, and we've sort of been slowly pulling in a network of people who are affected or impacted and want to send us a sample or just talk to us about it.
Okay, fantastic. Let's see. And this was a question I had. Do you see black goo in landfills that just accept construction and demolition debris?
I haven't in just C&D landfills, although I've seen them in more industrial waste landfills, particularly ones that are taking waste that had been solidified to pass a paint filter test.
Okay, and I know we've used the descriptive term that this black goo grows, but it sounds like that if it grows, it's not in the microbial sense of growth, but that we're seeing accretion or deposition, or I'm not quite sure those are the right terms either. But the question is, if it's not growing where we find it, then why do we think it's collecting in the sumps and the gas wells versus getting pumped out before it collects?
Well, I think there is a microbial component to this, because I think that is altering the surfaces and making them more amenable to creating sticky masses that glom together. But I do think the quiescent environment that it's, when something hits the bottom of a landfill, it's on top of the liner in the leachate collection system, things aren't moving as fast. It's in a sump, it's sitting there. These polymers, which may be flowing with the leachate, have an opportunity to essentially precipitate, to stick together and form a mat on the bottom. And I do think this microbial component could be a real clue to essentially something that's facilitating that occurring.
Yeah, we're seeing it in our sumps, right? So it is sitting there.
Yeah.
And then you have flow and then flows. So if it's mobile, that's kind of how you get that accumulation.
It seems common in dead spots, right? Where the flow is low. And at sumps too, I see sumps are a real problematic area, right?
Okay. And then one more question before we depart. I know we're running up on time here. Has anyone seen any kind of correlation with temperature in terms of temperature having any effect, whether that's perhaps attributing to the black goo phenomenon, or perhaps even as a way to help mitigate black goo formation?
We've seen it more mobile at higher temperatures. But Laura, Brittney, I think you've
I mean, I
experienced
just
some
think
hot
like
water.
on your timeline of events, like it is more frequently seen in the summer.
That's when we pull pumps, right?
Yeah.
So it's, we have seen it. Yeah. I mean, you look at the body temperature of a landfill, right? It does change, but I mean,
it's more stable than the surface.
yeah, exactly.
Yeah.
So, and I mean, because that's the best time to do the maintenance. So we pull those pumps at that time. And unless there's any irregularities that we're seeing or issues happening, we're following that time. So I can't definitively answer that question. Yeah, I wish I could. But.
Gotcha.
I could add, we've seen this in conventionally operating landfills where temperatures are normal, whatever that is, and that at ETLFs as well. You know, I've got one that's running 150 C, and we've got black goo in there, so it's hot, right?
Wow.
Gotcha. Okay. Great. Well, thank you all so much for your time today. This has been great. Brittney, Laura, Craig, your expertise has been very helpful in helping us all understand this challenge a bit more. Hopefully we can check in with you in another year and you can tell us that there's no more black goo anywhere at landfills or something. but barring that, we would love to bring you back again for another update on how we're dealing with the challenge. And so thank you all so much for your time. One last point here before we jump off. If you have not seen it, next week we'll be having another science session, this time on the topic of PFAS for resource management and talking about policy and legislation impacts related to PFAS. So please join us next Wednesday, the 23rd for that. Just as a reminder, you will be getting an email after this with information you can do your certificate of completion. And thanks for your participation. Thanks again to our speakers. You'll have a great day.
Thank you.
Yeah, thank you so much.
Yeah, thank you for having us. Yeah.